Hello, my name is Luther Evans, and I'm very excited to share my summer experience participating in the 2016 Bioscience project in neuroscience research. What captivated me to join this internship was my deep interest for how the brain operates and the cutting-edge technologies that are intertwined with modern scientific research. This presentation video will elaborate and provide more context, visualization, and explanation on my research topic that cannot be conveyed completely on my presentation poster, which is my primary means of displaying my research on analyzing gene expression patterns in associative learning. The starting point began with exploring potential neuroscience research topics relating to a mental disorder such as autism, depression, bipolar disease, schizophrenia, or a behavioral process such as learning to memory, aggression, addiction, etc. I had a profound interest in studying behavioral process of learning and memory with a focus on associative learning. So my ultimate purpose of my project would be to analyze gene expression patterns for brain regions involved in the encoding, storage, learning, and retrieval of memories, essentially to identify a candidate gene for this behavior. The primary data was obtained from the Allen Brain Atlas, which is a repository that combines neuroanatomy with genomics via gene expression maps. The gene expression data for the relevant brain regions, hippocampus and amygdala, were analyzed to uncover patterns, correlations, and networks. This information assists research efforts in behavioral neuroscience by producing results that generate new data-driven hypotheses for experimental study. Differential search option provided the gene expression data for the hippocampus and amygdala brain region as compared with the rest of the brain from various donors. In the microarray data from the Allen Brain Human Database, mRNA is copied into cDNA and then labeled and hybridized to an array that has all human genes. Typically, there are two different sample types that are used for comparison, the sample under study versus the control. The cDNA for each of these samples is labeled in a different color, then the two experiments are compared. Certain genes are expressed more in the subject under study than in the control, and these will be shown in red. Other genes will be expressed less, and these will be shown in green. If the expression is the same in both samples, it will be shown in black. For the data wrangling, the gene symbol and the fold change columns from each of the 4 probes files were also downloaded from the Allen Brain Atlas. The raw dataset was claimed before analysis because for each dataset there are usually replicate entries of the same gene in probes with unknown variables. The bioinformatic tool VENI compares lists and identifies what's common and different between the lists of genes. The results from the VENI common genes will be used for 2 purposes. 1. Graphing the top 10% of highly expressing genes, and 2. Cluster analysis. Graph the top 20 expressing genes for each dataset, 4 graphs in all. The highlighted genes that are common to all 4 donors were distinguished. The gene symbol and the fold change were also used in the Python and bioinformatics statistical analysis. Fundamental statistical parameters such as mean, variance, relative variability, and standard deviation were used when describing and comparing distributions. Behaviors and neurorelated processes are conserved. That's why I looked for genes that are highly expressed and conserved across all 4 donors. To tease out genes that are directly related to this specific project, I performed a technique that is used often in bioinformatics to gain information on large datasets known as clustering or grouping. The cluster analysis program I used was Database for Annotation, Visualization, and Integrated Discovery, also known as DAVID, which partitioned my gene lists based on different criteria related to function. From these results, I got a small number of genes to use as a hook to the protein network analysis, string, where I filtered out other genes that may be relevant to associative learning. The genes were clustered using Go terms. Go stands for gene ontology. This is a standard descriptive vocabulary used for classification. The lower p-values are important. They indicate statistical significance, whether to reject the null hypothesis, which in this case would be that the clusters occurred by chance alone. Also, they indicate how well the genes in the category clustered. GeneWeaver was also used for finding more information about genes of interest. GeneWeaver integrates data from many sources and across species as well. It will search all of these experimental databases for the occurrence of these two terms.